Okay, so today we're going to talk about DNA replication. And so, what is DNA replication? So, DNA replication is the process of basically making a new strand of DNA from a pre existing strand of DNA. We're just kind of making a photocopy of it. And so, what are some reasons why we need to go through DNA replication? Oh, wait, we already know. It's called mitosis. So, DNA replication occurs within the S phase of interphase in mitosis. We also know that we do see this DNA replication occur because cells are damaged. We need to repair them. We need to replace pre-existing cells because they've just met their life expectancy, and because we're growing and we need more cells. So we go through mitosis. And so we're trying to make an exact copy of the DNA found within the nucleus so that we can go through our normal everyday process. DNA replication is extremely important because it can lead to mutations if it's not done correctly. If those mutations are done within our normal autosomal cells, those do not get passed on to our offspring. However, when chromosomal mutations occur because of DNA replication that happens within meiosis, then those germ cells do pass that mutation on to our offspring. So it's really important that we do this correctly and that we form two identical strands of DNA. DNA must be the exact same every time. And so there's only 3 steps to DNA replication, but let's look at a DNA molecule before we go through this. So remember that a nucleotide is made up of a phosphate, which are you little green things, a sugar, a nitrogen base, and then they're bound together by hydrogen, but we have these oxygen in there. I want you to note the fact of the oxygen, because when we start talking about DNA replication, we see a 5' to 3' and 3' to 5'. But what on earth does that mean? Well, our sugar is called deoxyribose. On each one of our sugar molecules, we have a visible oxygen showing. You'll notice that the oxygen, when it goes 5' to 3' is on the top, and when it goes 3' to 5', it's on the bottom. That's so that those binding points here for our hydrogen bonds that hook our nitrogen bases together are in the right spot for that. And so with this, we have we have to have them bind in a certain way. So if you notice, they're counting 1, 2, 3, 4, 5. That's occurring in the direction of what way it is from the oxygen. So it first starts with the binding spot to that nitrogen base, and it will go all the way down to our oxygen being that last 5th spot. Right before the oxygen is our number 5 carbon, and where it's bound to that. And so that's how we get that 3' to 5'. It really has to go with the direction of this sugar and how it's attached to the phosphate in relationship with the nitrogen base, which becomes some important information as we go into our next 2 slides. So step 1 is simply unwinding the DNA strand. Remember, the DNA strand is a double helix. And so if you notice, we still have our 3' and 5' up here. And we use a protein that's really important. This protein is called DNA helicase. So we know it's a protein because it has ASE at the end. And so DNA helicase is breaking those hydrogen bonds that's found between the 2 nitrogen bases, exposing each side so that we have 2 separate sides, or what we consider a Y shape, or for the replication forks. And then we see that we have a thing called DNA polymerase here, and that is going to bind. But we'll look at that in just a second. So here's a fun little graphic of our enzyme just ripping those hydrogen bonds apart to make those replication forks. It's a very, very quick, fast, efficient process. So step 2 is adding those complementary base pairs in. Remember, adenine will always bind to thymine, thymine binds to adenine, guanine to cytosine, cytosine to guanine. Always back and forth with those nitrogen bases. And when they come in, they're not just bringing a nitrogen base, but they're bringing a nucleotide. So that is a nitrogen base, a sugar, and a phosphate group. It is the job of DNA polymerase to bind the new nucleotide to the pre-existing nucleotide, so the complementary nucleotide. So let's look what's happening. So DNA helicase is doing its job. It's making those replication forks. But we can see that we have our two strands. We have our leg strand and our leading strand. So if you notice with our leading strand, we're starting with a 3' binding to a 5' going this way. With our leg strand, we're starting with a 5' and we're moving in the opposite direction of that. So we're kind of moving backwards. And so we'll talk about Ozark fragments in another section, but what's happening here is they're bringing those nitrogen bases. So we have this polymerase, this DNA polymerase is coming in. It's looking at this primer that's already been set, and it's like, okay, here we go. It's binding 3' to 5'. So we always have the 3 to the 5. And again, that's the location of the sugar, of where those carbons are located at within the sugar. So we need to bind those in those spots so that the nucleotides line up in that specific sequence. And so that's the job of the DNA polymerase. We're not really worried about the leg and leading strand for this next test, but we do need to know that the DNA polymerase is bringing in that complementary nucleotide in to bind to the pre-existing nucleotide. And then what happens is that DNA polymerase is not just bringing it in, but it's proofreading it too. It's making sure it didn't make any errors. And that's really important because when we're done, we are going to have a complete DNA strand here. And if we notice, our thymine is to our adenine, our cytosine is to our guanine, and it's all the way down. And we have our deoxyribose and our phosphates, and we have these hydrogen bonds holding all of this together. Remember, hydrogen bonds can reform and break really quickly. So we can have something like DNA helicase come in and break the hydrogen bonds to make a replication fork. And then we can have something like DNA polymerase bring in new nucleotides and reform those hydrogen bonds just as efficiently. Our new DNA molecule is made up of 1/2 of the old and 1/2 new. So we're always reusing or recycling that DNA. So it's really important that they stay within that. When we're done, we have 2 brand new strands, identical strands of DNA. And again, we do this in mitosis so that we can go through replication to replace old existing cells, and we do it in meiosis to make gametes. Again, in the S phase of interphase is when this occurs. And until next time, when we talk about gene expression.